Welcome, listener, to the Deep Dive. Today we're taking on a really important and complex topic, bipolar disorder. You've handed us this stack of fascinating sources. We're talking genetics, neurobiology, treatments, early life stuff, a lot. Our mission today,
Wow.
dive deep, pull out the key nuggets, and hopefully give you a clearer picture.
Yeah, and what's really striking, I think, is how all these different research areas, genetics, brain chemistry, they're really starting to converge. It's painting a much more integrated picture of bipolar disorder. We're moving beyond just the list of symptoms
Right.
towards the underlying mechanism.
Exactly, like putting together a really complex puzzle. Still lots of pieces missing, maybe.
Yeah.
So let's unpack this. Where do we start? What is bipolar disorder according to these sources? How is it defined?
Well, fundamentally, it's defined as a clinical syndrome, which basically means it's characterized purely by symptoms, what someone experiences and reports.
Okay.
We don't yet have objective biological markers, like a blood test or a specific brain scan pattern.
All right.
That definitively says this is bipolar disorder type 1 versus type 2 or something else entirely. No biomarker really differentiates the clinical subtypes based on biology.
So it's not like one single neat illness with one clear cause.
Exactly. It can look quite different in different people, and, other conditions can sometimes present with similar features, too, which raises a really important question, doesn't it? How do we even study something so complex and varied?
That's a great point. It makes interpreting the research tricky. Okay, so our sources dive deep into the brain itself, the neurobiology. Let's start with the chemical messengers, neurotransmitters. What's going on there?
Well, historically, research really zeroed in on neurotransmitter dysregulation, imbalances, but it's broadened out a lot since then. Now, the focus includes things like intracellular signaling, like the communication inside the cells,
Okay.
and mitochondrial energy metabolism, how the cells make energy, oxidative stress, which is like cellular damage or rust, and even neurogenesis, the brain's ability to grow new neurons or connections. So, for instance, GABA levels, that's an inhibitory neurotransmitter, kind of the brain's brake.
Yeah.
They've often been found to be lower during both depression and mania.
Lower GABA. Yeah.
And conversely, there's evidence for what's called a hyperglutamatergic state, basically too much glutamate activity. Glutamate's the main excitatory one, the accelerator.
Ah, okay. So less brake, more accelerator, especially in certain areas.
Yeah, particularly areas like the frontal cortex seem implicated.
So it's about that balance being off. Shift.
It seems so. And what's fascinating here is that mood stabilizers, common ones like lithium and valproic acid, are actually thought to work, at least in part, by decreasing those synaptic glutamate levels.
Really?
Yeah, which supports the whole hypothesis, right? That neuronal hyperexcitability, brain cells being overly active, is a key mechanism.
That makes sense.
Yeah.
Connects the treatment back to the proposed mechanism. And building on that, stress keeps coming up in these sources, a big factor. Which brings up that critical question. How do our actual experiences, how do they literally change our brain?
Right. The sources explain that our environment, both psychosocial and physical, can hugely impact our neurobiology and our behavior. It triggers this process called allostasis.
Allostasis.
Yeah, think of it as the body constantly adapting, trying to achieve stability through change, responding to demands. And as our experiences change our brain and our thinking, we are essentially changing our own neurobiology.
Wow.
An individual's internal balance, their sort of homeostatic set point, that can determine how vulnerable they are to stress-related diseases, especially if they have that genetic predisposition.
This is where it gets really interesting. You're saying our thoughts, our experiences, they can literally reshape our brain chemistry.
That's what the evidence points to. The brain is constantly adapting.
Incredible. So beyond neurotransmitters, what about other crucial processes like communication inside the cells?
Oh,
Cell health.
yeah. So we also see dysfunctions in those intracellular biochemical cascades. Think of them like complex domino effects inside the cell.
Okay.
And those problems, along with that oxidative stress we mentioned, and mitochondrial dysfunction, the energy problems, all that can impair neuronal plasticity.
Plasticity meaning?
The brain's ability to change, rewire, adapt, learn.
Got it.
And when that's impaired, it can lead to cell damage, even cell loss of brain tissue, which we see evidence for in postmortem studies and also in neuroimaging, brain scans, just as an example of the complexity. ATT, usually a signaling molecule outside cells, In high concentrations, it can actually become cytotoxic, toxic to cells,
Wow.
and pro-inflammatory.
Oh, wow.
But its byproduct, adenosine, tends to be neuroprotective,
Mm.
anti-inflammatory.
So it's incredibly delicate, a real balancing act.
It really is.
So with all these potential targets, what does this mean for developing better treatments?
It means translational research is just critical, absolutely vital. We need to build those bridges, take the basic science findings, and translate them into discovering novel therapeutic agents that can work in people.
Makes sense. Connecting the lab bench to the clinic.
Hmm.
Okay, we've touched on stress influencing neurobiology. Let's zoom in on the body's main stress control center, the HPA axis. What exactly is that, and what's its role here?
Right, the HPA axis, hypothalamic pituitary adrenal axis, it's basically the command chain for your stress response. Brain signals pituitary, pituitary signals adrenals release cortisol, the stress hormone.
Got it.
And dysfunction in this HPA axis is strongly linked to what's called neuroprogression in bipolar disorder. It seems to induce these persistent changes that can make future episodes more severe, maybe more resistant to treatment.
So the stress system itself gets kind of dysregulated over time.
That's the idea.
Yeah.
And one common finding is high basal cortisol levels, just higher baseline stress hormones, especially noticeable in the morning and often during manic phases.
So people with bipolar disorder might literally be sort of swimming in more stress hormones a lot of the time, even when things feel calm.
In many cases, that seems to be true, yes. The cortisol awakening response, that natural spike right after you wake up, is often exaggerated or augmented. And this dysregulation, this HPA axis problem, can stick around even during remission, during euthymia, and it can actually predict future relapses, both depressive and manic.
Wow, that's a powerful link. Okay, the sources also mention how stress affects specific brain networks. Can you break that down for us?
Sure. So there are these large-scale brain networks that work together. 3 that consistently pop up in bipolar disorder research are the salience network, or SN. That helps us detect what's important or relevant in our environment.
Okay, the notice this network.
Kind of, yeah. Then the executive control network, ECN. That's for planning, decision-making, focus.
You get things done network.
Right. And the default mode network, DMN. This one's more active when we're like mind-wandering, reflecting, thinking about ourselves or others.
Right. The daydreaming network, maybe.
Often called that, yes. Now, these networks are often shown to be affected in bipolar disorder, even without immediate stress. But stress interacts with them too. In healthy people, acute stress ramps up the salience network activity, helps you detect threats, cope.
Makes sense.
But in people with genetic risks, like close relatives of schizophrenia patients who share some genetic overlap with bipolar disorder, their salience network connectivity might actually be lower during stress.
Lower. So they're less able to detect or respond appropriately?
It suggests maybe an impaired detection or an inadequate response when under pressure, yeah.
And what about that default mode network, the DMN? How does stress affect that one?
Well, in healthy folks, acute stress tends to temporarily increase DMN activity, which might make you more prone to interference from your own internal thoughts and emotions.
Okay.
But for people who are vulnerable, if they can't properly downregulate the DMN after the stress is over, that might lead to increased rumination, getting stuck in those negative thought loops.
I see.
And that could potentially be a precipitating factor for mood episodes in bipolar disorder.
Okay, so we've got neurotransmitters, cell health, stress response, brain networks. But why are some people more vulnerable to all this? The sources really emphasize genetics. How strong is that link?
It's pretty strong. Twin studies consistently estimate the heritability, how much variation is down to genes, to be between 60% and 85%.
60 to 85%. That's really high for a complex condition.
It is. It tells us genetics play a major role.
But it's not like there's one single bipolar gene causing it all, right?
Definitely not. It's what we call polygenic, meaning many different genetic variations likely contribute a small amount to the overall risk.
Okay.
And these variants often have incomplete penetrance, meaning not everyone with the variant gets the disorder, and variable expressivity, meaning it can show up differently.
So it's complex, which raises the question, how on earth do scientists even find these contributing genes?
The main tool for that has been genome-wide association studies, or GYS.
GYS.
Yeah. These studies scan the entire genomes of thousands, sometimes hundreds of thousands of people, comparing those with bipolar disorder to controls. The largest GWRs for bipolar disorder so far identified 30 independent genetic regions, or loci, associated with the disorder.
30. Okay, and what kind of genes are in those regions?
They include genes involved in things like ion channels, which control electrical signals in neurons,
Yeah.
neurotransmitter receptors, where the chemical signals dock, and synaptic components, the machinery of the connections between neurons.
So genes related to how brain cells communicate and fire,
Yeah.
linking back to that hyperexcitability idea.
Exactly. It all starts to tie together.
Okay, here's where it gets even more layered. The sources talk about epigenetics. What is that?
Right, epigenetics. It's a fascinating field. It basically refers to mechanisms that can change gene expression, whether a gene is turned on or off, without changing the underlying DNA code itself.
So it's like a volume controls or dimmer switches for genes.
Yeah.
Rather than changing the wiring.
That's a great analogy, yeah. Think of them as little chemical tags or modifications on the DNA or associated proteins. And these tags can tell the cellular machinery whether to read a particular gene or ignore it. It allows our genetic potential to adapt, to some extent, to environmental signals.
So this is how the environment might interact with our genes to influence risk. This is the bridge.
Precisely. It's a key mechanism for that gene-environment interaction. Things like DNA methylation or histone modification, these are types of epigenetic marks. And what's really interesting is that these patterns can sometimes be inherited.
Inherited. The epigenetic marks themselves.
Some can, yes, but they can also be modified by things we experience during our lifetime, like environmental exposures, or, very relevant here, experiences like childhood maltreatment.
Wow.
So it offers this biological pathway for how life experiences can actually get under the skin and impact brain biology and vulnerability.
It is profound. Really changes how you think about risk. Okay, shifting gears slightly, but it connects. Mitochondria. The cell's powerhouses, they come up again and again. Why so critical here?
Well, the brain, the central nervous system, is incredibly energy hungry. It has huge metabolic demands.
Right, uses the disproportionate amount of the body's energy.
Exactly. So it's particularly vulnerable if the energy factories aren't working efficiently. And mitochondrial dysfunction is increasingly seen as a really key mechanism in the pathophysiology of bipolar disorder.
How strong is that link?
Consider this. People who have diagnosed primary mitochondrial diseases, they show up to a 20-fold higher incidence of bipolar disorder compared to the general population.
20 times higher. Wow, that's a massive link.
It strongly suggests problems with energy metabolism are deeply involved.
So it really is like the brain's batteries aren't charging right or holding their charge.
That's a very good way to put it. We see evidence of impaired energy metabolism. For example, sometimes elevated lactate levels in the brain, which suggests cells are shifting to less efficient ways of making energy. And postmortem brain studies have shown reduced activity of a specific part of the mitochondrial energy-making machinery complex in the prefrontal cortex of BD patients. And what's fascinating here is how this connects back to that oxidative stress idea.
The cellular rusting.
Yeah, mitochondria are a major source of reactive oxygen species, ROS, or free radicals. Normally, ROS have important signaling roles. But if there's an imbalance, too many ROS or not enough antioxidants to clean them up,
You get oxidative stress.
exactly. And that oxidative stress can damage critical cell parts, like the mitochondrial DNA itself, proteins, lipids.
So it's a double whammy. Less energy production and more damage from the process.
Potentially, yes. It can become a vicious cycle.
Are there any biomarkers, things we can measure, that support this mitochondrial link?
Yes, there are some indicators. N-acetylaspartate, or NLA, its levels reflect how healthy neurons and mitochondria are. And NLA levels are often found to be decreased in bipolar disorder patients.
Lower NLEA.
Though interestingly, some studies show NLA can increase with treatment, like lamotrigine.
Oh, interesting. A potential sign of treatment working at the cellular level.
Possibly. Another marker is BCL2. It's an anti-apoptotic protein, meaning it helps prevent programmed cell death. BCL2 levels have been found lower in the prefrontal cortex of BD patients post-mortem, and lower levels in the blood during a manic episode seem to correlate with how severe the mania is. Less protection, more severe symptoms.
Got it. So multiple lines of evidence pointing towards these energy and cell health issues. Okay, another concept that comes up is neuroprogression. What does that mean in the context of bipolar disorder? It sounds concerning.
It is a concerning concept, yeah. Neuroprogression basically describes this idea of a pathological rewiring or even deterioration of the brain that seems to happen over time in parallel with the illness course. So as symptoms worsen or recur, there might be underlying progressive changes in the brain itself.
So the illness itself changes the brain over time, potentially making it worse.
That's the hypothesis. It's like the brain structure and function are altered by the disease process.
What kind of changes are we talking about? What does it look like in the brain?
Studies show structural changes, things like neuronal loss, fewer brain cells in both cortical areas, the outer layers, and subcortical areas, deeper structures. For example, one study found that individuals hospitalized for a first manic episode showed smaller gray matter volumes in the prefrontal cortex on both sides.
Smaller prefrontal cortex.
Right.
That's key for executive function.
Exactly. And there's also evidence of early changes in white matter. That's the insulation around nerve fibers that helps signals travel fast.
Right.
Things like lower signal intensity in the corpus callosum, the big bridge connecting the two hemispheres, and reduced total white matter volumes have been reported. And if we connect this to the bigger picture, the clinical course, the number of mood episodes someone has seems to be the most robust factor linked to this deterioration.
So more episodes equals potentially more neuroprogression.
It appears that way. More prior episodes are associated with faster recurrences, more functional impairment, disability, cognitive problems, and even, sadly, an increased risk of dementia later in life.
Wow. That really highlights the importance of preventing episodes. And what's really striking here is how inflammation fits into this neuroprogression idea.
Yes, inflammation seems to be a really crucial mechanism driving or contributing to neuroprogression in bipolar disorder.
How so?
Well, we see elevated levels of inflammatory mediators, things like certain cytokines, TNF-alpha, IL-6 in the blood during mood episodes. And these levels are linked to how active the illness is and how it's progressing.
Okay.
There's also evidence suggesting the blood-brain barrier might be more permeable or leaky in BD patients, allowing inflammatory stuff potentially easier access to the brain.
The protective barrier is compromised.
Potentially.
Yeah.
And postmortem studies back this up too. They find signs of increased neuroinflammation, more activation of immune cells in the brain, like microglia, and also astrocytes and oligodendrocytes seem involved.
So the brain's own immune system might be overactive or contributing to damage.
That's a key area of research, and it raises an important question, doesn't it?
Which is?
Could controlling or reducing this inflammation actually help slow down or prevent neuroprogression?
A very hopeful thought. Targeting inflammation as a treatment strategy.
Yeah.
Okay, let's talk about cognition. We often hear about thinking difficulties. What do the sources say about specific neuropsychological impairments?
Right. Cognitive issues are a major concern. Patients often show significant dysfunction across a pretty broad range of domains. Things like executive function planning, problem solving, working memory, attention, sustained attention, focus, verbal memory, learning and recalling words, visuospatial memory, remembering locations or patterns, and psychomotor speed, just how quickly you can process information and react.
So quite widespread potential difficulties.
Yes. And importantly, these deficits aren't just present during mood episodes, depression, or mania. They can persist even during euthymia, during periods of remission or stability.
So even when someone feels relatively well, their thinking might still be affected.
That's often the case, yes. Which can significantly impact daily functioning, work, relationships.
But you mentioned earlier it's heterogeneous. What does that mean for individuals? Does everyone experience these cognitive issues?
That's a crucial point. While group studies consistently show these average impairments, there's actually notable heterogeneity or variability from person to person.
Okay.
When researchers use stricter criteria to define impairment, they often find that only a minority of individuals show really global, across-the-board impairment.
So it's not everyone, and maybe not in all areas for most people.
Exactly. Some studies even suggest there might be different cognitive clusters or subgroups. You might have a group that's globally impaired, another group that's largely intact, maybe some with very specific deficits, like primarily in processing speed or verbal learning or social cognition.
That really highlights the diversity within the diagnosis itself, doesn't it?
It absolutely does.
So what does this mean for how we should approach understanding and potentially treating these cognitive aspects? A one-size-fits-all approach probably won't work.
No, it's just we need a more personalized approach, and it raises an important question about how we even assess cognition in research. Most studies are cross-sectional, just a snapshot in time.
Right.
Which really limits our understanding of how stable these deficits are, or how they change over the course of the illness. Do they get worse with episodes? Do they improve with treatment?
We need the long view.
Exactly. We need more longitudinal studies following people over time, and maybe more sophisticated statistical methods too, things that can look at individual profiles, not just group averages. Techniques like finite, partially ordered sets or other individual level analyses. The goal is to map individual cognitive strengths and weaknesses, which could ultimately help tailor interventions or even stratify the illness better.
Okay, that makes a lot of sense. Moving beyond averages to individual patterns, let's circle back to something we touched on. This idea of kindling or sensitization. Our sources describe it as a key model for progression. Can you explain that?
Sure. The kindling or sensitization model basically suggests that repeated exposures, either to stressors or perhaps even to the mood episodes themselves, can lead to the brain becoming progressively more sensitive over time.
More sensitive, not more tolerant.
Exactly. Instead of adapting or getting used to it, the system becomes more reactive. Think of it like building a fire. The first sparks might not do much, but with repeated sparks, stressors or episodes, the kindling catches, and eventually it takes less and less of a spark to ignite a full-blown fire, a mood episode.
So earlier stress or earlier episodes literally lower the threshold for future ones, making you more vulnerable.
That's the core idea, yes, and it links strongly to early life experiences. Early life stress, particularly childhood maltreatment, physical, sexual, emotional, even chronic verbal abuse, is consistently identified as a significant risk factor for bipolar disorder.
How does it affect the illness?
It's associated with an earlier age of onset, starting younger, and often a more severe course of illness, more frequent relapses, more hospitalizations. Also linked to higher levels of ongoing psychosocial stress and more co-occurring conditions, things like substance use disorders, anxiety disorders, and various medical problems too. Allergies, arthritis, migraines, thyroid issues.
Wow. Even verbal abuse alone.
Yes, the sources emphasize that even verbal abuse can have a really pernicious, damaging effect on its own. It shouldn't be underestimated.
That's sobering, the long shadow of early experiences. And this is where it gets really interesting, almost sci-fi. Our sources suggest this sensitization, this vulnerability, might even be transmitted across generations. How?
Yeah, this is where epigenetics comes back in, but with an intergenerational twist. So, beyond just inheriting DNA sequence variations, parental experiences, particularly things like stress exposure, or maybe substance use like cocaine or THC mentioned in some animal studies, these experiences might alter those epigenetic marks. The methylation, the histone modifications, maybe microRNAs within their sperm or eggs.
Within the germ cells?
That's the hypothesis being explored, yes. And if those altered epigenetic marks are passed on, they could potentially influence brain development, stress reactivity, and overall vulnerability in the offspring.
So you're saying someone's biological parents' experiences, things that happen to them, could influence their child's risk, even if the child never experienced those things directly.
The emerging evidence, particularly from animal models but increasingly explored in humans, suggests that is a real possibility. It adds this whole other layer to understanding how vulnerability might be transmitted, a transgenerational epigenetic effect.
Mind-blowing. Does it mean it's deterministic, set in stone?
Not necessarily. What's also fascinating is some animal work suggests potential reversibility. For example, studies showed that if adult male mice are conditioned to fear a certain odor, and that fear is transmitted epigenetically to their offspring, if the father mice are then given extinction training to overcome that fear before conception, then the fear is no longer transmitted to the next generation via the germline.
Wow. So interventions could potentially break that cycle.
It certainly offers a hopeful possibility, though it's early days for translating that to humans.
Still incredibly powerful. Okay, we've covered a huge amount of ground. Genetics, epigenetics, stress, mitochondria, brain changes, cognition, kindling. Bringing it all together, what are the biggest implications for actually treating and maybe even preventing bipolar disorder?
I think the single most overwhelming message coming through all these complex sources is the absolutely critical importance of early intervention.
Early intervention.
Yes. The earlier treatment starts, especially after a first manic episode, the better the chances seem to be of modifying the long-term course of the illness, potentially preventing or slowing down that kindling-like progression, that neuroprogression we talked about.
And you mentioned something earlier about recovery.
Right. Some evidence suggests that if there are no further episodes after the first one, then cognitive function and even some of the brain abnormalities observed can actually normalize, return towards baseline within about a year.
That's incredibly hopeful. It really underscores the need to act fast and effectively after that first episode.
Absolutely. It's a crucial window.
So what does all this complexity mean for current treatments, like mood stabilizers, and where might future treatments be heading?
Well, current mood stabilizers like lithium, valproate, lamotrigine, they actually seem to have quite broad effects across many of these pathways we've discussed. They influence glutamate neurotransmission. They can affect mitochondrial function, intracellular signaling pathways like PKC and GSK3. They can reduce oxidative damage, promote neuroprotective factors. So they're hitting multiple targets, which might be why they work, albeit not perfectly for everyone.
Right. But research isn't stopping there. People are looking for more targeted approaches.
Definitely. Research is actively exploring new targets based on all this neurobiological understanding. And one particularly fascinating area is looking at mitochondrial modulators.
Targeting the energy factories directly.
Exactly. Compounds that might boost mitochondrial function or protect against damage. Things like coenzyme Q10. CoQ10, and acetylcysteine, or NEC, which we know has antioxidant, anti-inflammatory, and glutamate modulating effects.
NAC, right. Heard about that.
And even acetyl L-carnitine, ALC. There's evidence it's low in depressed adults who experience childhood adversity. And in animal models, it shows antidepressant effects, potentially through epigenetic mechanisms.
Interesting. Any downsides?
Well, with ALC, there have been some case reports linking it to manic switches in bipolar disorder, so caution is needed. But the idea of maybe a mitochondrial cocktail is being floated, combining agents, maybe something energy-enhancing with an antioxidant to get synergistic effects and potentially minimize side effects.
A cocktail approach. Makes sense given the complexity.
We also saw mentions of rapid-acting treatments gaining attention. Ketamine, for example. It's an NMDA receptor antagonist affecting glutamate. It's shown rapid antidepressant effects even within hours in people with the treatment-resistant bipolar depression.
Rapid effects are definitely needed sometimes.
And on the therapy side, mindfulness-based cognitive therapy, MBCT, shows promising results specifically for preventing depressive relapse in bipolar disorder. And it seems to work in part by improving emotion regulation skills.
So, pharmacological, mitochondrial, therapeutic. Lots of avenues. Given everything we've unpacked today, what stands out to you as maybe the most critical path forward? Where should the focus be?
For me, based on these sources, it really feels like the need for systemic change, particularly focusing on children who are at high risk.
High-risk children, offspring of parents with bipolar disorder.
Exactly. The statistics are stark. One source mentioned rates as high as 74% of these kids receiving a major childhood psychiatric diagnosis over an 8-year follow-up.
74%. That's staggering.
It is. And given what we know about early adversity kindling neuroprogression, interventions like family-focused therapy, FFT, which involves the whole family, and consistent monitoring of parental symptoms to ensure stability, these seem absolutely crucial, proactive prevention.
So really translating the research into practice for those families.
Yes. Bridging that gap seems like one of the most impactful things we could do. Moving from knowing this stuff to actually implementing it widely to offer these kids and their families a better future.
That's a powerful call to action. This deep dive has certainly been a journey, hasn't it? Through some incredibly complex, interconnected systems, from that subtle dance of neurotransmitters, the brain's energy supply, all the way to the huge impact of early life stress, and even these transgenerational epigenetic echoes. It's crystal clear that bipolar disorder is far, far from a simple condition.
Absolutely. And if we connect it all back to the bigger picture, it really underscores that there isn't going to be one single magic bullet cure or one easy answer. It's a multifaceted challenge. It demands a personalized, integrated approach considering all these factors. But the hope, I think, lies in continuing to unravel these intricate mechanisms, because the better we understand them, the better chance we have of developing more precise, more effective interventions down the line.
Absolutely. Understanding leads to better tools, better strategies. And as you, our listener, process all this information we've explored today, here's maybe a final thought to mull over, something to carry with you. If our life experiences, particularly those really early ones, can literally shape our brains, influence our vulnerability, and potentially even cast shadows onto future generations through these epigenetic changes, what responsibility does that place on us as a society? A responsibility to actively protect and nurture childhood development. Not just for the well-being of individual children, but for the collective mental health of entire populations, perhaps for generations to come.